The name of Charles Darwin's book that everybody knows, the abbreviated name, is On the Origin of Species. And how exactly do species originate? How do new species develop? Well, before we get into that, we have to talk about what exactly is a species. A species is a group of organisms that won't interbreed with similar groups in nature. So reproductive isolation is the definitive definition, biological definition of a species. And sometimes it's really easy to tell if they're different species. Cats and dogs, definitely different species. They don't interbreed with each other. What about Labradors and poodles? They're very different from each other, but yeah, they can interbreed with each other. They're different, they're the same species. On the other hand, without human intervention, can this Great Dane and this Chihuahua interbreed with each other? Would they interbreed with each other in nature? No. We have actually, through artificial selection, created new species. And in this case, it's called a clinal species, where we have extremes of variations. The extremes cannot interbreed with each other. So these are two different species of dogs. But what about things we only know from fossils? How do we know they're different species? How do we know they don't interbreed with each other? Well, we really don't. We can just rely on their structures. They're morphological species. If they're structurally different from each other, they're different species. For example, we have gorillas, Neanderthals, humans. It's pretty well agreed upon that humans and gorillas are different species, and they exist today, and they definitely don't interbreed. What about Neanderthals and Homo sapiens? Well, in the past, it was thought that they were different species. Then we found in areas where they lived together about 40 or 50,000 years ago or so, there were some intermediate, so maybe they did interbreed together. And whether they're different species or the same species, it depends on what scientist you talk to. So sometimes it's not quite so easy to define whether or not they're the same species. What about lions and tigers? Do they interbreed in nature? Well, they don't encounter each other in nature. So we do consider them different species. They are structurally different enough that they don't interbreed. And when we put them together in artificial situations, in zoos, they do interbreed, and hybrid is called a liger. But lions and tigers are still considered different species because they don't interbreed in nature. Lions occur in Africa, tigers occur in India and the southeast, South Pacific, in those areas, and they don't interact with each other. And this is one way that species avoid interacting with each other. It's called geographic isolation. In this case, it's large distances and oceans. In other cases, it might be something as simple as a river or a mountain. Sometimes similar species are isolated from each other based on timing. For example, there's a type of skunk called an eastern spotted skunk. They interbreed with each other in late winter. And then there's a western spotted skunk. They mate with each other during the fall. So even though these two species may live in the same area, they won't normally breed with each other in nature because their mating season is different. Now, what we talked about, geographic and temporal isolation, these are referred to as prezygotic isolating mechanisms before a zygote forms. And this figure shows what a zygote is. A sperm cell fertilizes an egg cell and you get a zygote, a single-celled embryonic stage. And that's later going to turn into an embryo and a fetus and a newborn. Some other examples of prezygotic isolating mechanisms include behavioral isolation. Behaviors keep different species from mating with each other. For example, we're mostly seeing on this slide fruit flies, sometimes also called vinegar flies. These are extensively studied for their genetics. They have 8 chromosomes, just a small number of chromosomes. We figured out their entire genome. They have a lot of different characteristics. And on the bottom left, you see what's referred to as a cladogram or family tree of them that shows the different species. Now, the different species are related to each other. And even though they kind of all look alike to us, there are some subtle differences. They can tell a difference. And you can see on the bottom right, little animation of a female in the front, and a male is following around behind her and doing a certain dance, and he's extending one of his wings, and this particular species has a certain type of mating dance. Now, on the top left of this slide, you can see fireflies. Fireflies emit light, and different species of fireflies will emit light at different frequencies. And the males will do a lot of flashing, and they will attract females. Females will get close to the males, and then the females will start flashing, and they'll mate with each other. And this shows different species of fireflies that have different frequencies and ways of flashing their lights. And there's one species that's kind of interesting. The female can adjust how she flashes her light. She will see a male of a different species flashing, and she will mate, or she will flash with a certain way to attract this male, and he'll come to her to mate with her, and then she'll eat him. So she will attract males from other species by manipulating how she flashes her light. And then when she sees a male of her own species, she'll flash in a certain way to attract that male, and then she'll mate with him. But again, this is another example of prezygotic isolation or behavioral isolation. Flashing lights. There's also mechanical isolation, where the male and female parts have certain rigid shapes, and they have to match up in order for those species to mate with each other. Essentially, his lock fits her key. And this is quite common in arthropods, in insects, for example. And this figure shows the male reproductive parts of certain insects. And there are certain species of beetles where we can't tell the difference except if we dissect their reproductive parts, the male parts. These days, we also rely a lot on genetics, but before we had access or knowledge about the genetics of these beetles, entomologists would dissect their reproductive organs and be able to identify the different species of these beetles. Otherwise, they look identical to each other. Now, the beetles can tell the difference, but we can't, not so much. So that's mechanical isolation, where the male parts and the female parts have to fit together in a certain way, otherwise they won't reproduce. Another prezygotic isolating mechanism is called Gametic Isolation, and essentially, if they're from a different species, the sperm cells and the egg cells simply can't find each other. Sperm cells have chemicals, or not chemicals, but proteins in their cell membranes that match up to chemicals that the eggs are releasing. Essentially, the sperm cells can smell and swim towards the egg cells and they find each other. But if they're different species, they won't interact with each other. And this happens in a lot of invertebrates where they have external fertilization, such as sea urchins. Sometimes mistakes occur. These prezygotic isolating mechanisms don't work. And a sperm cell of one species will fertilize the egg cell of another species, and you'll get a zygote. And then there are mechanisms that keep those species from being that successful. Sometimes the hybrids just plain don't live. For example, in salamanders, the top right figure, when one species accidentally mates with another, eggs are laid, and the hybrids just don't survive. The eggs don't develop. Sometimes the hybrids are infertile. I think a lot of you have probably heard about this one. If a horse and a donkey mate with each other, what do you get? A mule. And the mule is a lot bigger and stronger and tougher than a donkey. It's also tougher than a horse, which is better adapted to running really fast. So you get this nice, good working animal called a mule that has a lot of good characteristics we like, but it's not a successful mating. And there's also hybrid breakdown. The hybrids survive. They can reproduce with each other, but the offspring of these hybrids don't survive. And thinking back to Mendelian crosses that we did earlier, you have the P generation that produces the F1 generation. The F1 generation organisms reproduce with each other, and it's the F2 generation that doesn't survive. And this is an issue in cotton. So these different mechanisms keep one species from mating with another, keeps one species isolated from the other, and they can be used in the process of speciation to create new species. They don't intentionally create new species, of course, they're not intentionally used, but these mechanisms act to create new species. In speciation, one species splits into two or more species. And there are a couple of types of speciation. In general, one of them is called sympatric speciation, which we'll get into a little bit later, where two populations are physically interacting with each other, but they don't mate with each other. That results in two different species. I'll talk about that later. But now we're going to talk about allopatric speciation, where populations are physically separated from each other. For example, thousands, if not millions of years ago, a certain species of bird called a cormorant ended up on the Galapagos Islands. The Galapagos Islands are 600 miles off the west coast of South America, and they're pretty isolated. And these cormorants started living on the islands, and the islands, Galapagos Islands, don't have any land predators that would eat the cormorants, and their wings atrophied. Their wings got smaller. So on the left, this is a figure of a flightless cormorant. This is another species of cormorant that has normal wings. And the flightless cormorant, the ones that devoted more metabolic energy into growth and egg laying and less metabolic energy into developing wings and feathers on wings, they were able to reproduce more successfully than cormorants in other areas which really needed better flying techniques to escape predators or to hunt down their prey better. So this population of birds became isolated on the Galapagos Islands. They adapted to local conditions. Their wings got smaller, and a new species developed. And these were physically isolated from other species of cormorant. A couple of other examples. Antelope squirrels around the Grand Canyon. On one side of the Grand Canyon, there's one species. On the other side of the Grand Canyon, there's another species. In the past, the populations became isolated from each other, and these squirrels adapted to their local conditions and became different species over time. And there's a type of shrimp in the Gulf of Mexico and the Pacific across the Panama. They're somewhat similar to each other, but they are different species. In the past, sea levels were high enough that these populations can interact, and there was one species, and then later ocean levels fell, and the land of Panama rose up out of the ocean and blocked these populations from each other, and those populations, isolated populations, adapted to local conditions and became different species of snapping shrimp. And one type of speciation like this is called adaptive radiation, where you start out with one population and it produces many different species. And the Galapagos finches are a good example of this. The Galapagos Islands, the oldest ones, formed somewhere around 2 or 3 million years ago, and finches from South America ended up on these islands and adapted to local conditions. And then occasionally the finches flew to different islands and adapted to more local conditions, and on different islands the finches became different enough that when they did meet again in the future, they didn't reproduce with each other. So one species developed into many species. That's adaptive radiation. And these populations were isolated from each other on different islands, and sometimes different habitats on the different islands. And this shows South America, the Galapagos Islands, 600 miles to the west of South America. So it's well isolated from South America. And it shows the different islands, and it's difficult for these finches to fly from one island to the next. They're far enough apart, many miles apart, and it's difficult for them to fly from one island to the next. So populations of finches on one island are mostly isolated from populations on different islands. And even within one island, the environments can be different enough where finches living in wetter forests are isolated from finches living around desert-like area with cacti. And this shows a few different species of the Galapagos finches. So populations become isolated with each other, and over time, inbreeding becomes less and less likely as these populations diverge and become adapted to local conditions. And we've seen this happen within our lifetimes, especially in situations where we have imported species. For example, there are a couple of species of fruit flies called starch fruit flies and maltose fruit flies. And over time, hybrids between these fruit flies are less and less common. These occur in the Sierra Nevada mountains in the western United States. And, well, actually, this is a different organism. There's a type of flower called a monkey flower that's also speciating. We're watching it happen. In the Sierra Nevada mountains, there are pink monkey flowers that are pollinated mostly by bumblebees, and red monkey flowers that are mostly pollinated by hummingbirds. And they're adapting to local conditions, and they're becoming different species. Now, these examples of fruit flies and flowers in the Sierra Nevada mountains, these populations are close to each other. The individuals geographically can interact with each other, but for different reasons, they don't. And this type of speciation is referred to as sympatric speciation. And sympatric speciation, the populations are not isolated from each other. And one situation where this can occur quite often in plants, it can occur because of polyploidy. Remember that we are mostly diploid. Most of our cells have two sets of chromosomes. Our sperm and egg cells have one set of chromosomes, or haploid. But if there are 3 or 4 or more sets of chromosomes, they are polyploid. 3 sets of chromosomes is triploid, 4 sets of chromosomes is polyploid. And remember meiosis. We have 4 sets of chromosomes here, 4 chromosomes, and the S phase of meiosis occurs, and each chromosome is made up of 2 copies. And the chromatids crossing over might occur. During meiosis 1, the homologs separate. During meiosis 2, the chromatids separate. So that's normal meiosis. Now, what may happen is the parent species during meiosis the chromosomes fail to separate. So you may have some diploid pollen grains or diploid egg cells. And you may come across a different species that also this happened with, and their pollen grains or egg cells are diploid. They have a full set of chromosomes, creating all of the proteins and enzymes necessary for that cell to survive. And a zygote forms. It has 4 sets of chromosomes. It's tetraploid. And this tetraploid organism, it has a full set of chromosomes from one parent and a full set of chromosomes from another parent. It can produce all the proteins and enzymes it needs to survive. And the tetraploid survives and can be so genetically different and so characteristically different from either parent that this tetraploid is a new species. So polyploidy can result in instantaneous speciation. And then here's another example of polyploidy in strawberries. And sometimes these polyploid plants have certain characteristics we like. In the case of strawberries, they produce bigger fruits. And what can happen is a tetraploid individual can reproduce with a triploid individual or diploid individual and produce triploid individuals. This is where most of our wheat comes from. Most of our wheat is triploid. So this shows our triploid wheat. It happens to have really big seeds that we like. Another example of Sympatric Speciation occurs in a type of fly called an apple maggot fly. About 200 years ago, they fed on hawthorn plants. And you can see hawthorn plants towards the bottom of this slide. Then we started growing domestic apples in the United States. And these apple maggot flies moved into apples. They're an economically important pest. And now these apple maggot flies feeding on domestic apples are genetically different than the ones that are still feeding on hawthorns. We can measure these differences. There's a reduced gene flow in the fly populations that are feeding on hawthorns and the fly populations that are feeding on apples. They don't reproduce with each other as much as ones that are living on the same food. And are these individuals new species? Are we seeing speciation occurring? So another example of sympatric speciation, and this has been seen in a good number of insects where the insects, certain individuals move to a new food, they become better adapted to that new food, and they diverge from the parent populations that's still feeding on the original food. Now, speciation can occur at different rates. It can occur quickly or slowly, geologically speaking. They may form new species over tens of millions of years, that's fairly slowly. Or they may form a new species over millions of years instead, or maybe even hundreds of thousands of years. Geologically speaking, that's very quickly. And when they become species very, very quickly, that's called punctuated equilibrium. And again, they can form new species over the course of hundreds of thousands of years instead of millions of years. And looking at the fossil record, we've seen this occur over the course of 4,000, well, let me rephrase that. New species can develop over the course of 4,000 years to over the course of 40 million years. And if it's closer to 4,000 years, we would call that punctuated equilibrium. And if it's closer to 40 million years, of course, that's just very slowly new species developing. And on average, speciation takes place over the course of 6.5 million years. And basically what happens if the environment very rapidly changes, new species can very rapidly form. We talked about that earlier when we're talking about how mammals very quickly developed after dinosaurs became extinct 65 million years ago. Before 65 million years, there weren't that many mammal species, and they mostly resembled mice and squirrels. And then a meteor impact struck the earth and wiped out all the dinosaurs. And then mammals very, very greatly and quickly diversified. Punctuated equilibrium occurred with mammals.